Actually, first of all, I would like to thank Dr. Ahmed Atif Salam, the founder of the MGBS. Uh, Today, Dr. Anokye Bismark from International Institute of Tropical Agriculture, Pan-African University, Ghana, will give a lecture on MGBS series seminars. Dr. Bismarck gained his Master of Science in Plant Breeding and Genetics in October 2021, Bachelor's of Science in Agriculture Biotechnology in June 2018. He has like awarded many awards, like beneficiary of the West African Center for Group Improvement and Pan-African University Scholarship from 2019 until 2021. First class student in Bachelor Agriculture Biotechnology at Canos in 2018. Uh, he got like a best speakers in debate competition on are GMOs good for mankind in 2017. He's actually like a member of a lot of international scientific committees. Like he's a member in the American Chemical Society and Crop Science Society of America, the African Plant Breeders Association in 22, and he is like the executive member of the science club at Juju State College. Uh, Plus, he is like a member in bioinformative group and a lot actually of many scientific societies, actually international societies. So, Dr. Bismarck title for today, Identification and Validation of Single Nucleotide Polymorphisms for Marker-Assisted Selection of Genotypes with Increased Carotenoids, Hydrogen Cyanide, and Dry Matter Content in Cassava. So, Dr. Bismarck, please go ahead.
All right. Thank you so much, Dr. Ahmed, for having me. I hope I am audible.
Of course, you're welcome.
Hello. Okay.
Yeah, go ahead.
All right. Okay, like you said before, today my name is Bismark Anokye. I'm from Ghana. Today I'm delivering a lecture on the genetic diversity study for hydrogen cyanide, carotenoids, dry matter content, and definition of single nucleotide polymorphisms associated with the trait in cassava. That's many hot esculenta. So I'll quickly go on with my presentation. Malnutrition has been a growing problem among low-income earners, specifically in Asia, Africa, and some parts of the world, which affects over 3 billion people worldwide. And among these factors, vitamin A deficiency, which is known as a hidden hunger, affects millions of people. And the group of people that are susceptible to this particular problem is children at the age under the age of 5 years and pregnant women. In fact, there was a statistic that over 140 million children at below 5 years suffer from vitamin A deficiency. Out of these children, 450,000 people or 550,000 of these children go blind, and then half of them actually loses their lives as a result of lack of vitamin A. Pregnant women also suffer such problems. And then it is known that over 410.7 billion US dollars annually is lost due to blindness. Now, worldwide or globally, you see in this map, the global map, and that the issue is highly situated in Africa and some other part of Asia. Per the map demonstrated with red ink or the red writings indicate that the places where the disease is most prevalent in the world. And then you could see that Africa has a problem, and then part of Asia. Now, there has been so many interventions geared toward solving the issues of VAD, which is vitamin A deficiency. And among the supplementation and then biofortification. However, the first three former approaches, though, are very cost effective, but for some variety of reasons have failed to alleviate or completely do away with vitamin A deficiency. And then so biofortification, which appears or which is a long-lasting and cost-effective means in delivering provitamin A to children and even some women across Africa and other parts of the world, remains a feasible approach in solving vitamin A deficiency in Africa or in other parts of the world. There has been so many approaches now. When we say biofortification, that is introduction of nutrients varieties within staple crops, and then making them available for people to consume. There have been different biofortified crops like maize, potatoes, and then cassava. But for the sake of this research, we will focus more on cassava. And then why cassava? Realize that a variety of people, or large number of people that are suffering from vitamin A deficiency, large number of them depend on cassava for survival. And then so improving cassava with micronutrients such as vitamin A, iron, zinc, and other nutrients go a long way in solving vitamin A deficiency. And then so it is not only producing biofortified crop or cassava that is high in provitamin A. There should also be other factors that should be taken into consideration or into account. Factors such as the dry matter content, which is a large portion is starch, is a primary factor that defines adaptation of new variety. And one major characteristic of cassava genotype is also the general presence of cyanogenic glucoside, such as hydrogen cyanide, that upon hydrolysis or enzymatic hydrolysis produces the free hydrogen cyanide, which is known to negatively affect human health and then human health. So now there is a need that even as we focus on producing biofortified crop or crops that are high in provitamin A, we also take the dry matter content and then the hydrocyanide content into consideration. In 2018, there was a report in Uganda that a small community in the evening consumed a flour which is made from cassava. And I think about 50% of the they were taking and cassava that contain a lot of hydrocyanide content. So why cassava? Like I said before, we have about 800 million people deriving their direct energy from cassava daily. And then out of these people, over 500 million people of them live in sub-Saharan Africa, which is the SS. And then this accounts for about global cassava production, because we're looking at the global cassava production, this region, which is sub-Saharan Africa, we produce about 60% of cassava of the total world. And it's not actually stored in the ground as food. And then traditionally grown and processed by women for consumption and the earning income. And then so we realized the importance of cassava and why people in sub-Saharan Africa and other parts of the world, they largely depend on cassava. And then so we try to biofortify cassava and then making this quality trait also available so that as they consume the cassava, they will also get these traits that are also contained in the cassava. So for the sake of this presentation, I'll be focusing on the presence of total carotenoid content, dry matter, and then hydrocyanide content in cassava. Now, there have been so many breeding approaches and breeding objectives. As an institute, IITA, since 2014, Harvest Plus with alliance with other institutions have come out with an approach to biofortify certain crops, which is maize, cassava, and then potatoes. And then so there has been varieties that has already been released with additional traits, and then they are also adapted to certain regions. And then so when you look at the biofortified cassava that has been released, that is the IBA 011368. And then it is high in provitamin A content. It's also resistant to CMD, which is the cassava mosaic disease. And then it also has very good quality for gari and fufu with high dry matter content. And then I add cassava varieties that are also adapted for other processing. processing and the market. So these are some of the varieties already available on market for sales. And then so this is the cassava pipeline. This is the cassava pipeline process, how we're able to generate the whole varieties, that design. So you sit down, you analyze, okay, which part of or which qualities in cassava do I want to produce or do I want to produce to the market? So it's more of interactive breeding and then foresight breeding. And then so you interact people more, you let know the need and the market. Then you do trait discovery. How then do you do trait discovery? It's more of evaluation of germplasm source. So you have varieties. So you do evaluation, trait validation, inheritance, and molecular discovery. So that is where this process comes in. So in the process of my lecture, I will focus more on this area. So if you go ahead, that's the stage 2, trait development. Stage 3, you do some crosses and then screening. Stage 4, you do early testing, late testing, pre-commercial testing, product registration, and then finally you release your variety. This is the cassava pipeline that we have designed. And then in designing these things, you also have to make some tools available so that you could increase selection. You could increase gain to selection. So we make this equipment available. When you come to IITA, there are so many equipment that are even used in the lab, some high throughput phenotyping approach and genotyping approaches. You could see we have tablets that we use to pick some phenotypic data on the field. And then when it comes to screening process like Provitamin A or determining the yellow intensity of cassava, we have tools like the iCheck carotenoid, which can be used even on the field. We have the spectrophotometer and the HPLC. These are all available. And the other tools also, we also have some other descriptors that are also used in taking data and then also try to improve cassava varieties. Now, in breeding, whenever we are breeding, I want us to take this particular equation into consideration. Now, whenever you want to check response to selection, number one, you consider the accuracy. That is a better phenotyping approach. You consider the size of your population and the germplasm diversification. Whenever there is diversity, then you have options in selecting a particular variety that best suits your breeding program or breeding objectives. In taking these 3 factors into consideration, you also consider time. Cassava breeding has mainly due to phenotypic recurrent selection. And then when you look at phenotypic recurrent selection, we are talking about 7-8 to minimize the time that you use to develop a particular variety. That is why this particular lecture becomes very important. Now, like I said before, in developing cassava variety, you begin with cross-block source population. You generate your seedling nursery. After 1 year, you must trial, advanced yield trial. Trial the UIT, then you go ahead. Hello, can you hear me? Hello. Dr. Mohammed.
Please. Yeah. Hi. Yeah.
Can you hear me?
Yeah.
Okay. All right. Thank you.
Okay.
All right.
Go ahead.
All right. Okay. Thank you so much. So due to the time that is required to develop a variety, that is about 8 years. Then looking at the advantages that comes with the phenotypic recurrent selection, that the phenotypic recurrent selection, it's slow due to the annual growth cycle and low multiplication rates. Conventional breeding also for new varieties a lot of years, about 10 years, like I said before, and the breeding complexities associated with the trait involved. Then there is a need to complement phenotypic recurrent selection with molecular approach. And then so you do so that subsequently you'll be able to get molecular markers in your screening process. So application of high throughput genetic markers in screening systems such as SNPs could fast track the improvement of cassava for desirable traits. And then so in molecular breeding, we mainly focused on two basic approach, which is genomic selection and then marker-assisted selection. Some also term as marker-assisted breeding. For the sake of this lecture, I will focus on marker-assisted breeding. So there are so many molecular markers that are available. So depending on your choice and your objective, you select a particular molecular marker. But in this lecture or in this research, we use a single nucleotide polymorphic markers for this particular research. Now, molecular markers are actually combine classical plant breeding with the tool and the discovery of molecular biology and genetics, more specifically to use of molecular markers. So there are classical molecular markers and then the molecular biology. Now, the use of markers in breeding cycle. So in breeding cycle, you have your population, you have your variety, you do your DNA extraction, and then obviously you use your markers to select the trait that you want. Now, for the properties, why we choose molecular markers over normal phenotypic recurrent selection, it's because of the high level of polymorphism. It's able to give us the differences, the codominant inheritant nature of markers. And then it's also usually ambiguous and designation of alleles. And then some other properties, it is also high reproducible. Whenever we choose SNPs, we are sure that the trait that we are improving will be able to improve it within a short time. A single nucleotide polymorphic markers, a marker system based on SNP provide an extra advantages. And due to the low genotyping cost, number one, the abundance in the genomic, the biallelic nature of SNPs amenable for high throughput detection format and then platform. Now, this advantage of SNP call for their application in crop improvement, specifically in cassava breeding. So you begin with your SNP discovery, the SNP that is associated with a particular trait. So you begin with it. You use, in this research, we'll use a GWAS, which is genome-wide association study, to identify our SNPs, and then we actually did further screening to select the putative SNPs, and then we focus on the validation. We validated these traits because for them to be considered as Mendelian genetics, there is a need for multiple validation in different genetic background, both technical and biological validation. And then so you have to validate this SNPs so that you will be used in marker-assisted selection process. So to facilitate the effective utilization of the functional markers in marker-assisted selection, for efficient selection, these potential SNPs have to be converted to the uniplex allele PCR assay, which is the CASP. That's a competitive allele specific PCR. So this is what we used for this experiment. For their widespread use and reliable as markers in molecular breeding, validation in multiple backgrounds, I said before, and independent population was conducted. So this is the entire summary of the whole process, the use of the next generation data processing, and how you'll be able to identify your putative SNPs to be used in marker-assisted selection. And then so with this research, the research was conducted in the Institute of Tropical Agriculture and then University of Ibadan in two different fields, but in the same region. And then we conducted the laboratory process or the laboratory analysis. Actually, the leaf samples were taken to Intertech, Australia, for the genotyping process, which the data was later sent for further analysis. So the laboratory analysis, such as the hydrogen cyanide analysis, the dry matter determination, as well as the total carotenoid content determination, all these things was conducted in IIT Nigeria, Ibadan, and then so that is it. So out of all 435 cassava genotypes from two different populations were used. I used one population which was at a seedling nursery stage, which was a harvest plus seedling nursery, and then the second population was from University of Ibadan, Nigeria, which was at the AYT stage, which is advanced yield trial stage. The seedling nursery was laid down in an augmented experimental design, which checks. And then for the second population, 2 meters, that of the AYT was 1 by 1 meters. All cultural and then agronomic practices were observed. in the genotyping, we used trait-linked SNPs for this particular, which were associated with 3 different traits. And then so we consider we have dry matter, which for the dry matter, we have 3 different markers for dry matter. The carotenoid content, I have 4 different, and then for hydrogen cyanide, I use 2 different SNP markers. Now, the favorable alleles for each marker is indicated there, and then that of the unfavorable allele. So when you go through these publications by Rabi 2020, Udo 2017, and the Obona in 2020, and this research was conducted in Cornell University. When you go through them, you're going to see all these markers and then how they were generated. So all these papers are available. So I want to use a pointer. Now, this is how the entire research was conducted. We have our cross on the field. Then after some times, we collected the leaf sample for genotyping. So this was used for the genotype. And then we carried them to Intech for analysis or for genotyping. Now, for field evaluation data, we collected field evaluation data such as the CMD severity and then other traits. And then at harvest, we also harvested the crop while we also considered the weight. In the laboratory. Realized we did laboratory sample. This is the sample that we prepared for the total carotenoid analysis. For the total carotenoid analysis, we used 2 different approaches, which is we use the spectrophotometer, and then we use the eye-check device. The eye-check device has an advantage of screening a very large population within a very short time. For the hydrogen cyanide analysis, we use the enzymatic hydrolysis using the auto analyzer to analyze the hydrogen cyanide content for the population. And then we also considered the dry matter. So the phenotypic data and the genotypic data were made together, and then we did a trait association analysis. The allele detection was based on fluorescence resonance energy, which is transfer quencher cassette, allowed by allelic discrimination of known SNPs. That is how the SNP basically work. So this is the workflow. This is how your SNPs were discovered using the CASP. So basically, it is cost effective than the multiplex. Now the price for the CASP is, I think, it's between $15 per essay. So it's really cost effective. And then the shorter turnaround time to receive the result. In fact, when your data is taken or your sample is taken within 1 week, your results is in. So that is the summary of the whole process. Now, the CASP actually contains 3 main components, which is the purified DNA, your 2 allele specific primers, and then a common reverse primer. And then sample taken is actually a minimum of between 5 to 10 nanogram of extracted DNA sample. It's required for the method to function properly. So the DNA sample is purified by adding a mixture of chemicals to the buffer solution. And then you continue with the process, which is in the manufacturer's guide. So finally, realize that your data, your SNPs is generated, and then you then have your data. So when your data is brought to you, and then you export your data in the SNP view, this is how it appears. You are going to see 3 different clusters. One allele, and then the heterozygous, and then the alternate allele. And so you are able to tell that if on your plate, this genotype, which is in the first one, is it heterozygous for that particular locus, you are able to tell. And so directly from there, you can then select. If you don't want to continue with any yield data, you can select and you continue your screening process. Now, when this data was taken, we made all the data available, and then for phenotypic data, we visualize the data, and then using histogram to check the distribution of it. And then we also use blue were obtained in the linear mixed model using the LIMP package R. And so this equation was used. This equation where the Y was a vector of observation, and then the B stand for the block with IF effect. The C stand for the check that was used for the experiment, and then while the TK genotype stand for the genotypic effect, and where the A stand for the random residual error. And then so the genotypic, the raw HAP map was actually generated, and then it was imported into Taxel to calculate the various SNP summaries, such as the polymorphic information content, the MAFs, which is the minor allele frequency, and then the heterozygosity as well. So the principal component analysis was performed to estimate the genetic structure of the cassava clones that were used for the experiment. Phylogenetic tree was also constructed using the phylogenetic and the evaluation. And then all this was done using R software. A box plot was also used to visualize the allele substitution effects based on observed phenotypic effects. So the ANOVA was also estimated, and the linear regression estimates the proportion of the variation explained by the markers. I also conducted a correlation between the objective marker and then the phenotypic data that were taken. I also adapted a selection index to select superior genotypes that will be advanced at the AYT stage, that is the advanced yield stage, and then at the seedling nesting stage we selected this one so that they could be used as potential parents for further trials. Thank you. So this data for the diversity study was generated. So here we have the two population, the advanced trial population and the seedling nursery population. And for the MAF, which is the minor allele frequency, range between 0.45 and 0.11 with an average of 0.2. And then at the seedling nursery, you could see 0.00. And then I will explain why this was observed. But then generally, you could see that the minor allele frequency for this particular data, it was really high. That of the gene diversity ranged from 0.19 to 0.50 at the AYT, and then the second population was 0.0 to 0.49. So the PIC, which talks about the informativeness of the markers, range from 0.17 to 0.37 with an average of 0.29. This genetic diversity parameters tells us that the markers that were used for this population were highly informative. Determination of genetic variation in breeding population also facilitate the identification of useful genetic divergence imperative for cassava population improvements. The gene diversity observed here was less than the mean gene diversity mean diversity, and this suggests a form of outcrossing or heterozygote excess, which in contrary to expectation under the Hedy-Weinberg equilibrium in the natural population. The two populations studied per this parameter showed moderate to high genetic diversity, and this suggests the availability of useful and unique allele, and that could be exploited in cassava improvement program. Secondly, I also conducted principal component analysis to determine the genetic structure of the population. The total amount of variation explained by the first principal component was 39%. Sorry, I should supposed to be here, but I think I skipped me. The first and the second principal component were also to explain about 22.3% and then 16.8% respectively of the total variation observed. You could see that there were 3 distinct clusters. Now, here I added the parents of the seedling nursery, because they also provide us with information about how the seedling nursery or the seedling nursery population is performing. And then so there was 3 distinct clusters observed here, but there was no distinct pattern along the 2 principal components based on the genotypic background or the source. Now, this indicates a low genetic differentiation. Yeah, this indicates low genetic differentiation. The cluster pattern of the clones along the PC axis reflects an intense genetic or intense exchange of genetic background. I also conducted a cluster analysis or the phylogenetic analysis, and then it grouped the population into three main groups. The estimate of the cluster membership that conveying the entire genotypes into three main groups, as it has been demonstrated here. We have group 1, which is very small. We have 15 of the populations, 15 of the genotypes. Group 2, we have 88, and group 3, we have 257 of the members. Now, per the data that was taken, the phenotypic data, members in cluster 1 had low hydrogen cyanide content and low total carotenoid content and low to moderate dry matter content. That is this group. Now, the second group, that is the cluster 2 members, generally high phylogenetic potential genotype with high carotenoids and high dry matter content. Then the genotype consists of low, moderate, and then high TC, dry matter, and then HCN content, which is the group 3 members. And so that is what we also observe with this cluster data here. I also performed some what is it called, broad sense heritability. I know someone will say, why did I not conduct a narrow sense heritability? Now, broad sense heritability was conducted because this particular experiment was not repeated and it was not conducted in different regions. And then so I estimated the broad sense heritability. And then the broad sense heritability for the eye check, which is the Provitamin A carotenoid, was very high, which is 0.93. This indicates that this trait is highly heritable. Now, for the TC chart, that's the use of normal color charts to also determine the yellow intensity of the cassava. The broad sense heritability was 0.77. The dry matter was very low, which was 0.2, and the ACN was also 0.68. Now, the end user preference, market value, acceptability of cassava genotype for root quality are related to their physiochemical composition. Now, the broad sense heritability was estimated to know the genetic effect of the phenotypic value. A high broad sense heritability denotes the ease of improving traits and hence can be selected at the early stage of cassava breeding program because it's controlled by mainly genetics. Now, before that, there has been a speculation and there are many publications about the negative correlation between total carotenoid content and that of dry matter content. Now, in this experiment, in the chart here or the matrix here, the A is that of the AYT stage, which is the first population, and then the B here, it's the seedling nursery trial. Now, between dry matter and that of the dry matter content and then the total carotenoid content, it was observed to be negative for the seedling nursery, the AYT. Why a positive correlation was observed for the second population, which is the harvest plus seedling nursery. And then going forward, I will explain why a positive correlation but very low was actually observed in the harvest plus seedling nursery. Now, the correlation among the traits also provide information on the relationship that can be explored for improved crop performance. Genotypes with a high dry matter content have a lot of white starch, and we tend to dilute the intensity of pigmentation. So that is why usually high dry matter genotypes will often have low total carotenoid content as a result of the presence of the white starch in the genotype. And the positive association between the dry matter content and the total carotenoid content could be as a result of the continuous recombination in the recurrent selection scheme imposed on the harvest plus seedling nursery population for increased total carotenoid content level and then dry matter content. So that is why we could see that there was a positive correlation in the IIT or harvest plus population, but a negative correlation observed in the AYT stage, which was a University of Ibadan clones that were generated. Now, a positive correlation observed between the eye check and then that of the AYT is actually due to the fact that genotypes with yellow roots are commonly found in the Amazon basin and then coastal area, where highly cyanogenetic lines are often preferred. So that is one factor. We also determine the quality of the nitrates linked with the markers across the two populations. So once you have your data, where I observed a very high call rate between 96 to 100% in the markers we use for the screening. Now you could see from A that they produced three distinct clusters. Three distinct clusters were observed. Now in the B, you only saw one cluster, and then in the C, you only saw two clusters. Ideally, if the population was segregating, we're supposed to see three distinct clusters. So it tells you that in B and in C, the particular marker was not polymorphic. So the two homozygous and then the heterozygous cluster were observed for all the markers except the markers S1, 22, and then S1, 19, and that of S12, 24 in the seedling nursery population. Now the S1 and then that of the S1, 22, and the S1, 19, this marker is associated with total carotenoid, while this one is a dry matter content marker. And then it's only distinguished the homozygote and then that of the heterozygote in the seedling nursery and that of the progenitors. Now, we also consider the performance of the markers linked to the total carotenoid, dry matter, and the cyanogenetic potential using box plots. So the first box plot you are seeing, it's the performance of the total carotenoid link SNPs in the AYT, which is the advanced yield trial stage. And then we could see a significance among the genotypic classes. Now, the favorable allele for this particular loci is AA, and the unfavorable allele is CC. And then you could see a high significance between the genotypic class, each individual bearing the 2 copies of the favorable allele and then a single copy of the unfavorable allele. So it tells you the contribution of this particular loci to the yellow intensity of the cassava. And whilst though the others could not really give us a significant difference. Now, the various loci contribute to total carotenoid among or between the genotypic classes demonstrate the importance of additive gene effect in the expression of carotenoids in cassava roots. Now, the second one is the allelic effect of total carotenoid content markers on the seedling nursery population, which is the IIT or the harvest plus population. Now, here was only one genotypic class that was observed in this particular population for this particular loci. And then it also explained. Now, phenotypically realize that all these populations, they were yellow. So it's explained that this particular trait for this particular loci is fixed within the population, and for that matter, the loci is not segregating. So the marker, which is the S122, predicts high total carotenoid content among the genotype in the two populations than the remaining markers, while the locus, the S2 or the S8, 83 predicts high total carotenoid content in the AYT, which is the previous one, for the favorable allele. So, but this S122 is known to be on chromosome 1, which is tightly linked to the phytosyntase gene, which is a key associate enzyme in carotenoid biosynthesis pathway. We also did analysis for that of the dry matter using the two populations. And then the dry matter, there was no significance for this one and then this one, but marker S119 was a bit higher, and then it could be able to differentiate those individuals bearing the favorable allele and those individuals bearing unfavorable allele. At the same time, in the first population, that is the harvest plus seedling population, we also found out that the loci for the dry matter, which is S119, is also linked with the phytosyntase gene on same chromosome 1. And then so it's actually brought our attention to one major discovery, why there's always a negative correlation between dry matter and then total carotenoid content. Now, in rabbit, reported the combination of these two suggests a strong physical linkage between the two loci for carotenoid content. And now, this is a continuous recombination of the selection imposed on harvest plus nursery and then that of dry matter. That is why we see only one genotypic class for both the marker for carotenoid and that of the marker for dry matter. Now, also go substitute. Now, the favorable for this particular marker is GG. This one is AA. So the two markers realized that the hydrogen cyanide, the toxic hydrogen cyanide within this population was very low, although few populations or few genotypes in this of the favorable allele. And then this because low cyanide content observed will actually help us to produce genotypes of superior varieties that have high carotenoid content as well as high dry matter content. So we also considered the effect of the markers linked to the total carotenoid content, and this same marker that is associated with the phytome synthase gene for that of total carotenoid content was highly significant. It was highly significant, and it accounted for 27%. This one loci accounted for 27%, and that of in the seedling nursery, it accounted for only 0.17% of the total variation in the root yellowness intensity observed. Now, the significant effect of this same marker, that is S122, in the different genetic background indicate the crucial role of the marker or its crucial role in the phytome synthase gene in the increased accumulation of total carotenoid content in cassava roots. I also determined the correlation effect between the predicted and the observed traits. And then the predicted model for the total carotenoid hydrogen cyanide content were also tested for their accuracies. And so this is what we did. This is the first population at the advanced yield trial stage and that of the seedling nursery stage. And then we could also see that the predictive value from the markers and the observed quantitative value were subject to correlation analysis. And this is what we are observing here, a significant positive correlation between the estimated total dry matter content, hydrogen cyanide content, and that of the total carotenoid content, and the predicted values by the markers in the two population was also observed, and the relatively low, realized that low R values were also observed in the population, though the P values we observed was very significant. Now, this tells us the importance of this loci in contributing to total carotenoid content, dry matter accumulation, and that of hydrogen cyanide accumulation in cassava roots. Now, I would like to pause here and then explain further the discoveries that we found in the course of this research. Now, for the marker S122, the uniqueness of marker S122 is this marker is on chromosome 1, and this marker is highly or slightly linked. It's tightly linked to the phytome synthase gene. It's about 572 kilobase of the phytome synthase gene. And then this marker play a major and a crucial role in carotenoid biosynthesis. So, and then this dry matter marker is also found within the same locus. It's also found within this same locus. Now, previous research by Rabi in 2017 reported the genomic coagulation of these two major loci suggests a strong physical linkage between the two loci for total carotenoid and that of dry matter. Now, before there was a speculation that there could be a pleiotropic effect where one gene may influence both traits because of the coagulation of the two loci on the chromosome 1 for both traits. When the gene is upregulated in one trait, it is downregulated in the other trait. Hence, she suggested further work should be done. But then this research is telling us today that now, irrespective of the allele that is present, being it a favorable allele AA for total carotenoid, when you continue doing the breeding and then improvement, If this trait is present and the unfavorable trait, unfavorable allele for total or dry matter content is present, you are likely to have a low dry matter content and then very high total carotenoid content. And then so attention should be taken or caution should be taken in during crop improvement program, so that when one is trying to improve these traits simultaneously, you will be able to improve for the total carotenoid, the favorable allele for total carotenoid, and then you also take the other allele for the dry matter content, but not the unfavorable allele, because at this point, it is the unfavorable allele that we are seeing here. So the percentage of the total phenotypic variation explained by the total dry matter, being it negative or positive, is dependent on the allelic substitution at the linked carotenoid biosynthesis at dry matter gene. So this is what I was trying to explain. Now, in RIT, these markers, these SNP markers for these different traits. Now, Provitamin A, these markers are then commercially available. That of the CMD resistance is also available. Dry matter content is also available, and then this CGMA resistance marker, they are all available to be used for marker-assisted selection process. Now, to conclude with the whole thing, based on the genetic diversity analysis that we did, and then the data we generated using the minor allele frequencies, the PIC now, and then the cluster analysis as well as the phylogenetic tree depicts a sufficient genetic variation exists among the two populations. Although in the AYT population, which is the seedling nursery population, which is the harvest plus seedling nursery, appears some traits for dry matter, and then that of hydrogen dry matter, and then total carotenoid appears to be fixed. Now that is the point theory. Now the favorable allele A for this this is favorable allele S1, and then that of the favorable allele for dry matter in the seedling nursery population appear to be fixed within the first population, where I was able to also select superior genotypes. That is the top 15 genotype, which combine high total carotenoid, which is above 19 microgram. And then the dry matter ranges from 30 to 40%, and then low hydrogen cyanide, which is below 60 milligram per 100 grams, were also identified. And then they are now being evaluated for further analysis. I will end by saying fixation implies that a trait, being desirable or undesirable, controlled by major genes, can be fixed through proper breeding procedure and effort in population improvement. I would like to acknowledge these personalities and institutions for their privilege and also being very supportive during or in the course of this research. And I also like to thank ASIUT Universities, the organizers, for making it a point and also honoring my invitation or taking me to be part of this particular presentation. I don't take it for granted. I'd like to thank you so much and thank each and everyone for being part of this presentation. Thank you so much.
Thank you very much, Dr. Bismark, and thank you for your informative presentation. Of course, cassava is the third largest source of food carbohydrates, of course, after rice and corn. And cassava is a major staple food in the developing world. I forget to tell everyone, Dr. Bismark, he's a PhD candidate, and of course, wish you all the best with your degree. So now, anybody has like a questions or wanna ask anything? Okay, so there is one. Okay, so okay, yeah, go ahead and ask a question.
Yeah, can you start right now?
Yeah, go ahead.
Yeah, I'd like to thank Bismarck for his nice presentation, and I'd like to ask, is there any crosstalk between the biosynthesis of carotenoids and the cyanogenic components or not?
Okay. All right. Thank you so much for that. So there was an association. I observed a more positive association between carotenoid content. I don't know if on the presentation where I actually brought the correlation metrics. On the correlation matrix, there was a positive correlation, and then I could explain why most of the yellow cassava or yellow root genotypes also have a little bit high cyanogenic potential. Now, it's cassava is Latin America origin, and then usually in these areas where the yellow root was discovered.
Yes. My question is
used. They prefer beta cassava. And then so during the introduction and then that of improvements, realize that they will always consider those ones genotypes with high cyanogenic potential. But because for them it's in this part of our world or in Africa, because we usually cook cassava and then we process the cassava. So basically the hydrocyanide or the HCN content becomes minimized at the point where we are eating. That is how it is helping us. But basically on my report, realize that we observe a positive correlation between HCN and that of the total carotenoid content.
Okay,
Yeah.
so yeah, do you have another question?
Yeah, it means if we can decrease the cyanogenic components, this will increase the carotenoids or not, or there is no.
Okay, all right. So there is a possibility. This is the reason why I also used a population which is at the seedling nursery stage, and then I harvested it at 17 months and 7 months, sorry, because at that stage it is known that the HCN content is very high, and then so if you are at this tender stage or at the seedling nursery stage where no selection has been imposed on the population, if you're able to get a genotype that has a very low hydrogen cyanide content, even with a high total carotenoid content, then you are sure that at the end of the day you are getting a genotype or a clone that has very low hydrogen cyanide to the barest minimum with high total carotenoid content.
Okay, I have another question, if it's possible.
Okay, yeah, go ahead.
Do you think biofortification with carotenoids can increase the content of beta carotenoid, or this is unuseful?
Okay, all right. Thank you so much for your question. Well, from correlation analysis that we have done, realize that the beta carotene, it's actually directly the intensity is the yellowness intensity has a direct and then a very high positive correlation with that of the beta carotene content. And then so when you use the HPLC, you realize that though there are so many components of the total carotenoid, but the ones that appears to be very high, the color intensity is very high, will always have a high beta carotene content. And then so by increasing the total carotenoid content, there is more than 95% probability that you are increasing beta carotene content in the genotype.
It means if we use beta carotene as a supplement in the growth medium, this will increase the.
So that is why basically.
Yeah, the content of the total carotenoids in the roots.
Please, can you come again? I didn't get your question. Or
yeah, please repeat your question. Yeah.
it is not a question. I'd like to say,
Okay.
if we add beta carotene as a supplement in the growth medium, it will increase the content of the total carotenoids in cassava. Is it right?
Oh, like in vitro? Are you talking about in vitro approach? Your line comes breaking.
Uh-huh.
Hello.
Could you please repeat it again and summarize what you wanna explain?
Yeah, if we add beta carotene as a supplement in the growth medium, for example, in the soil or in the hydroponic system, this will increase the total carotenoid in cassava or not?
Oh, that's no. In it won't. It may not be. It will not work because this is a genetic factor has been accumulate more of the total carotenoids. However, in this stage, realize that it is actually a genetic factor. Like if you clearly see some of my results, you realize that some of the genotypes didn't have the favorable allele. The SNP at the chromosome 1, which is the S1, which is linked with the phytium synthase gene, which is a main key enzyme that is responsible for yellowness intensity in crops. And then so if you don't have the traits and then just applying the beta carotene like that, it may not work. It may not increase the total carotenoid within your media.
Nice presentation, and thank you for nice discussion.
Yeah, thank you. Thank you for your question. So is there
Thank you
any
so much
other
for having me.
questions? Please don't be shy. If you wanna write it down in a chat room, I can read it for you. So let me check the chat room here. Actually, people thanks you, Dr. Bismark. Salim say thank you. Dr. Shahira El Musallami say thanks. Nada Fathi Hamada say thanks. Unknown user say excellent session. Yeah, so Razaz Muhammad say thank you. Agro Banjuko say thanks. I have a question. I am a plant protection person, and I'm
Yes, sir.
curious. Yeah, and I'm curious about disease and insect pests. So,
Okay. All
what
right.
is the most common disease or insect pest of cassava
Okay.
plant?
All right. Okay. Thank you. So in sub-Saharan Africa here, the common disease we have on cassava is mainly the cassava mosaic virus, cassava mosaic disease, which is vectored by the Bemisia tabaci, the adult whitefly. Yeah,
Uh,
so so they are capable of transmitting the virus to the cassava because of the way their eating habits. They are polyphagous eaters, and then so as they eat, and then they are able to transfer the virus. But then basically because here most farmers acquire their planting materials through their neighboring farmers, so you realize they intend picking up the diseased cuttings and then with an attempt transferring the disease to their farms. And then so I even have conducted I did one in vitro approach. That's where I was able to also produce clean planting materials virus free. That's using the meristem approach and then that of heat treatment. So I made the thing sprout at a very elevated temperature of between 40 to 45 degrees Celsius, where after sprouting, I acquired the meristem. So it is believed that replication process is paused or is impeded at that elevated stage, at elevated temperature. So once I generated them, I cultured them in vitro, I were able to generate and then do some virus indexing. We're able to generate clean planting materials. Yeah, so somewhere in southern Africa and Eastern Africa, you see that of the cassava brown streak virus very prevalent around these regions.
Okay, yeah, thank you. Let me go back for the in the chat room. There is Dr. Rehab Hafiz. She's like assistant professor of genetics, botany department, faculty of science, Cairo University. Thank you for your excellent session and excellent informative presentation. So, any other questions here? I think.
Yes, I have Muhammad.
Okay, yeah, Doctor. Yeah, our founder of the MGBS, Dr. Ahmed Atif. Yeah, go ahead.
Thank you, Bismark, for the really very interesting and incentive. My question is, are you going to like convert the good markers for CASP marker to be validated in different genetic background? Because it seems that you have really very good signals in genome-wide association study. So it will be nice really if you convert these markers to CASP, try to validate it.
Yes, yeah. Yes. So for we have converted these markers to CASP.
Okay. Nice. And is it published work or still?
Hello, Doc.
Is it published work? Did you publish the results?
Hello.
I mean, yes.
Yeah. Is there?
No. It's.
Yeah, go ahead, Dr. Bismarck. Dr. Ahmed asked about publications. Is there any publications from your work processing?
So I've provided my I've provided email. Yeah. So so most of these markers, a majority of these markers have been published, and then you see them in there's this thing I am providing with you now. One author, that's Rabi et al. 2020. You find some of the markers there. And then for that is that of the total carotenoid content and dry matter content, but that of the HCN markers, you find it in Ogbonna. This research was conducted in Cornell University, Ogbonna, Alex Ogbonna et al. 2020. Alex Ogbonna et al. 2020. So you find the markers, you find these markers available. And then we also work with Intertech, which is in Australia. Intertech in Australia. So they are able to, once you send your samples and you tell them that, okay, I want you to use.
Hello.
We lost connection.
Okay.
I think we lost his connection.
Okay, so yeah, we will actually, we would like to thank Dr. Bismarck for his time and for excellent presentation. Dr. Bismarck, he left his email address in the chat room. So if you have any further question, please go ahead and send him email about your questions. So finally, thank you very much for your attendance, and I wish you all the best. And please tune with us for the next Sunday with excellent presentation, and keep with us in the MGPS seminars. Thank you, Dr. Ahmed Atif. Thank you, everyone, and see you next week. Okay, thank you. Bye-bye.